Good afternoon, I am here with my biology project, and today I'll be talking about Piezo I protein and the gain of function mutation. The normal function of this protein is simply to sense mechanical signals and trigger intercellular responses. The category for this protein will be a receptor protein. The reason I chose this category is because this protein will react to mechanical stimuli happening in the body as a sensor, where now it will begin to open a channel to allow the flow of calcium and sodium ions. The subcellular location is in the plasma membrane. The type of cells that are contained when it comes to this protein are non-sensory tissue cells, and the organisms that this cell is found in is animals, including us as humans. This protein will become more active as a result of this mutation. The reason for this is due to the increase of the concentration of calcium ions when it comes to this protein. Now, regarding the original structure, here's the original structure. You see here that the water bottles are the calcium ion flow. They're the channels for the calcium ions to flow. At the ends are the functional groups, and these are the blades of the structure. The functional groups are carboxyl, amine, and hydroxyl. These three are highly reactive. This is hydrophilic due to the reactivity of their nature, and the charge is neutral, and the size will be relatively small compared to other amino acids such as tyrosine. Due to the presence of the hydroxyl group, the hydrogen bonding interactions can take place. Furthermore, due to its polarity nature, it can also participate in hydrophilic interactions to mix with aqueous solutions. The main functional groups in the mutated structure will be amine and carboxyl, but now the hydroxyl is substituted with a nonpolar group, which will now make this hydrophobic due to the nonpolar group taking place. The charge will still remain the same and be neutral, and the nonpolar group is relatively a small size. This mutated protein can no longer participate in hydrogen bonding like the original can due to the absence of the hydroxyl group, which makes it less reactive. Instead, it will participate in hydrophobic interactions, clustering with other molecules to avoid aqueous environments such as water. The effect that this has on the protein is simply that it will increase the calcium ions. As far as the functioning of the cell, it will have a tendency to alter and reduce iron metabolism. And at the functioning of the organism, it can systematically affect the organism. And this will be only due to the biological process that takes place when it comes to this protein, such as metabolism itself. Some of the limitations for my model, well, here is the mutated, the second model for the mutation. The calcium ions are here. You can see the increase in calcium ions to represent the mutation. Some of the limitations on my structure, well, the actual structure itself, in terms of the protein in the human body, is very dynamic. And it needs constant shifting to go on, simply because it's vital for proper ion channel functioning. Also, another thing with limitation with my model is that it doesn't represent the actual shape and orientation of the protein the same way as it does in the complexity of the human body. And so that is pretty much it for this presentation. Thank you.